Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The year 1983 resides in the historical memory of medicine as a period of profound uncertainty and escalating fear. A mysterious and devastating illness, then known as acquired immune deficiency syndrome, was systematically dismantling the immune systems of specific populations, leaving clinicians with descriptive observations and a desperate vacuum where a causal explanation should be. Amidst this crucible of scientific urgency, a small team at the Pasteur Institute in Paris, led by Luc Montagnier and including Françoise, Barré-Sinoussi and Jean-Claude Chermann, published a paper in the journal Science that would fundamentally alter the trajectory of the epidemic. Their work, titled Isolation of a T lymphotropic retrovirus from a Patient at Risk for Acquired Immune Deficiency Syndrome AIDS, presented a meticulously constructed, albeit cautiously phrased, argument for the existence of a novel retroviral agent. This essay will undertake a direct, section-by-section evaluation of the internal claims, methodologies, and conclusions presented within this seminal 1983 publication. It seeks to reconstruct the logical architecture of their discovery, assessing the strength of each piece of evidence as it was laid down, and to understand how, through a convergence of virological techniques, the authors built a compelling case for a new pathogen that would soon be known as the human immunodeficiency virus. The analysis will adhere to the paper's own progression, exploring the intellectual and technical scaffolding that supports one of the 20th century's most significant biomedical discoveries. Section 1. The conceptual framework and strategic patient selection. The opening paragraphs of the Montagnier Group's paper establish the intellectual landscape and the central hypothesis guiding their investigation. The authors situate their work within the existing paradigm of human retrovirology, dominated at the time by the human T-cell leukemia virus, or HDLV, which was known to cause a form of adult T-cell leukemia and possessed a tropism for the same general class of lymphocytes that were conspicuously depleted in AIDS patients. The core claim here is one of analogy and strategic divergence. They proposed that AIDS, a disease characterized by the depletion of T lymphocytes, might be caused by a related T lymphotropic retrovirus. The evaluation of this initial claim reveals its profound strategic insight. By framing the search in this way, they could leverage the established toolkit of retrovirology, including the essential assay for the enzyme reverse transcriptase, which is the biochemical hallmark of this viral class. The selection of the patient, codenamed BIU, is presented as a deliberate and critical choice. The patient exhibited persistent generalized lymphadenopathy, a condition understood as a possible prodrome, or precursor state, to full-blown AIDS. The implicit argument is that in this earlier phase, the patient's immune system would still be actively combating a viral agent, making viral isolation from a lymph node biopsy a more promising endeavor than attempting it from a patient in the terminal stages of the disease where the target cell population and the virus itself might be scarce. This decision reflects a sophisticated clinical and virological intuition, suggesting that the peak of viral replication and diversity might occur before the complete collapse of the host's immune defenses. The claim of the patient being at risk for AIDS was both the clinical description and a research strategy, positioning the investigation at the most opportune moment in the disease's natural history to capture its etiological agent. Section 2. The methodology of viral isolation and propagation. The paper proceeds to detail the techniques for culturing cells from the patient's explanted lymph node tissue. The central technical claim of this section is the successful short-term propagation of a viral agent through the co-cultivation of the patient's lymphocytes with T lymphocytes from healthy blood donors. The methodology itself is a testament to the technical state of the art in the early 1980s. The authors employed T-cell growth factor, the substance now known as interleukin-2, To stimulate and sustain the proliferation of the T lymphocytes in culture, this was a critical component, as these cells are typically difficult to maintain in vitro. The claim of successful viral propagation is supported by two primary observations. First, the authors report the detection of a cytopathic effect, meaning visible damage to the cells, which appeared in the cultured lymphocytes after a period of incubation. This effect was transmissible, as the supernatant from the initial culture, when added to fresh lymphocytes from a healthy donor, could induce a similar cytopathic effect. This observation provided the first biological evidence of a replicative, cell-damaging agent present in the patient's tissue. The second, and more definitive, piece of evidence was the detection of reverse transcriptase activity. The evaluation of this methodological claim reveals its foundational importance to the entire paper. Without a system to grow the virus, however transiently, none of the subsequent characterizations would have been possible. The reliance on primary lymphocyte cultures, while successful, also hints at the technical fragility of the discovery the virus they were isolating was actively killing the very cells needed for its production, creating a difficult race against time in the laboratory. The reported decline in both reverse transcriptase activity and cell viability after a few weeks underscores this challenge and explains why immediate and multifaceted analysis of the culture was paramount. Section 3. The biochemical evidence of reverse transcriptase activity. This portion of the paper presents the biochemical data that firmly identifies the isolated agent as a retrovirus. The core claim is the unambiguous detection of reverse transcriptase enzyme activity in the supernatant of the infected cell cultures. The authors provide specific details about the assay conditions, noting that the enzyme showed a preference for the template primer, combination of polyadenylate oligodeoxythymidylate, and required the presence of magnesium ions for optimal activity. This biochemical fingerprint, particularly the magnesium preference, was significant. It provided a point of comparison with the known human retrovirus, HDLV, whose reverse transcriptase was reported to have a similar cationic preference. The authors present a graph showing the kinetics of the enzyme's activity over time. The activity appears around day 15 of the culture, peaks, and then diminishes, a temporal pattern that correlates directly with the observed cytopathic effect and the decline in viable cells. Evaluating this claim shows it to be the linchpin of their argument. The reverse transcriptase assay was the accepted gold standard for detecting retroviral replication. Its positive result elevated their findings from the observation of a non-specific cytopathic effect to the specific identification of a retrovirus. The detailed reporting of the assay's biochemical characteristics was a display of rigor, demonstrating that the detected activity conformed to the known properties of retroviral polymerases. While the magnesium preference was similar to that of HELV, it was a general characteristic of many such enzymes and, by itself, was insufficient to prove or disprove identity. The true weight of this claim was its powerful confirmation of the retroviral nature of the transmissible agent they had cultured from the patient with lymphadenopathy syndrome, providing a solid biochemical foundation upon which all other claims would be built. Section 4. The morphological characterization by electron microscopy. Having established the agent's biochemical identity as a retrovirus, the paper moves to its physical visualization to electron microscopy. The central claim of this section is that the observed viral particles possess a distinct morphology. It differs from the known members of the HDLV family. The authors describe budding particles at the membrane of the infected T lymphocytes, a classic feature of retroviral replication. They provide electron micrographs as direct visual evidence. Critically, they detail the morphology of the mature virions, describing them as having a dense, eccentric core that was sometimes cylindrical or cone-shaped. This description was of immense importance. The established morphology of HDLVI and HDLVI, which belonged to the oncovirus subfamily, was typically characterized by a more spherical, centrally located core, the assertion of a morphologically distinct, cone-shaped core was a powerful piece of evidence suggesting that the virus from patient BIU belonged to a different retroviral group, likely the lentivirus subfamily. Lentiviruses, like the visnovirus in sheep, were known for causing slow, progressive diseases and exhibiting this particular morphology. An evaluation of this claim highlights its subtle yet profound impact. In an era preceding rapid genetic sequencing, morphology was a key taxonomic tool. The visual evidence presented in the micrographs directly supported the argument that this was a novel agent. This claim was direct, albeit implicit, challenge to the hypothesis that AIDS was caused by a variant of HDLV. The images themselves became iconic pieces of data, providing a tangible face to the otherwise invisible agent. The quality and interpretation of these electron micrographs were crucial. They offered a structural argument for the virus novelty that was independent of, yet complementary to, the biochemical and immunological Section 5. The immunological evidence for a novel virus. This section of the paper delivers what is perhaps its most conclusive argument for the novelty of their isolate. A core claim is that the virus, which they provisionally named lymphadenopathy-associated virus or LAV, is antigenically distinct from HELVI. The authors detail a series of immunological experiments to support this assertion. They used specific antibodies, including a monoclonal antibody directed against the major core protein of HDLVI known as P24, which has a molecular weight of 24 kilodaltons. When these antibodies were used in an indirect immunofluorescence assay on cells infected with LAV, they failed to detect any reaction. This indicated that the major proteins of LAV did not share the key epitopes, or antibody recognition sites, of HDLVI. Furthermore, they tested serum from the patient BIU. This serum reacted strongly with LAV-infected cells but showed no reactivity against cells infected with HDLVI. This was a crucial reciprocal finding. It demonstrated that the patient's immune system had generated antibodies specific to the new virus, and these antibodies did not cross-react with the known human retrovirus. Evaluating this claim reveals it as the definitive stroke that separated LAV from the prevailing candidate virus, while the reverse transcriptase activity and morphology were highly suggestive. The immunological data provided a clear, empirical line of demarcation. The statement of antigenic independence was a direct refutation of the idea that the isolate was merely a variant of HDLV. It strongly implied that they were dealing with an entirely different serotype, and likely a new species of human retrovirus. The use of both specific anti-HDLV antibodies and the patient's own serum provided a robust, two-pronged immunological argument that was difficult to dismiss. This section was a masterclass in comparative virology, using the tools developed for one virus to prove the existence of an. Section 6. The synthesis of evidence and tentative conclusions. The discussion section of the paper serves to synthesize the different lines of evidence into a coherent, albeit cautiously worded, conclusion. The overarching claim is that the authors have successfully isolated a new human T lymphotropic retrovirus from a patient at risk for AIDS. They methodically reiterate the evidence the virus could be propagated in T lymphocytes. It possessed reverse transcriptase activity, it had a unique morphology visible under the electron microscope, and it was immunologically unrelated to HDLVI. The cumulative weight of these independent lines of evidence forms the basis of their argument. The authors then advance a more speculative, yet critically important, claim regarding the virus' potential etiological role. They propose that this virus, LAV, is likely involved in the pathogenesis of AIDS. They base this on its isolation from a patient with a disease prodrome, its tropism for T lymphocytes, and its cytopathic effect on these cells in culture, which mirrors the T-cell depletion seen in AIDS patients. They also make a prescient observation about the virus target cell. They note that the T-cell subsets that supported viral replication were enriched for cells bearing the T4 marker, now known as the cluster of differentiation 4 or CD4 marker. This suggested, correctly, that the T4 positive helper T cell was the primary target of the new virus, providing a direct mechanistic link to the specific immunodeficiency observed in the disease. An evaluation of the concluding section reveals a skillful balance between scientific confidence and appropriate caution. The authors claim the isolation of a new virus with certainty, supported by their robust data. They propose its role in AIDS more tentatively, framing it as a strong possibility, requiring further investigation in other patients. This careful rhetoric was strategically necessary in a competitive and high-stakes research environment. The proposal of T4 cell tropism was a remarkable insight that would be validated and become central to the entire field of HIV risk. Conclusion: the 1983 paper by Montagnier, Barré-Sinoussi, and Chermann stands as a monument of methodical scientific inquiry. A deep evaluation of its internal claims demonstrates that its persuasive power emanates from the convergence of multiple, independent lines of evidence. Each section builds upon the last, constructing an inescapable logical case. The authors began with a sound strategic premise, selecting a patient at an optimal stage of disease to test a specific retroviral hypothesis. They successfully employed the demanding techniques of primary T-cell culture to propagate a transmissible agent. This agent was then unequivocally identified as a retrovirus through the gold standard reverse transcriptase assay. Its physical form, visualized by electron microscopy, presented a morphology distinct from the dominant retroviral candidate of the era. This morphological uniqueness was then given definitive support by immunological experiments that demonstrated the new virus antigenic independence from HDLVI. The synthesis of these findings led to the confident claim of a new human retrovirus, LAV, and the carefully articulated, yet ultimately correct, hypothesis of its causal involvement in AIDS. The paper's enduring significance lies in its rigorous, multifaceted approach. It did not rely on a single observation but on a consilience of evidence from biology, biochemistry, morphology, and immunology. While the scientific and political battles over priority and nomenclature would continue for some time, the claims advanced within the four corners of this publication constituted the fundamental scientific breakthrough. The paper provided the first tangible identity for the agent of a devastating plague, furnishing the essential tools for diagnosis, research, and the eventual development of therapies, thereby altering the course of modern medicine and public health.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.